Welcome everyone to the Rumen Health webinar series brought to you by Fibro Animal Health. Thank you so much for joining us and taking time out of your busy day to be with us today. As you can see, it is a series. Sorry, my screen has naturally frozen up. Here we go. It is a Rumen Health webinar series, and so we do have a couple opportunities to be able to learn more in this series. And those two webinars are coming up soon, one in 2 weeks, so May 20th, and that will be with Dr. Greg Penner with the University of Saskatchewan. And then the next one will be June 3rd. June 3rd is with Dr. Jenny Jennings from Texas A&M University. And you can follow the link there on your screen at www.academy.tahc.com to go ahead and register for those webinars in advance. We'll share that link again at the end of the webinar so that you'll have that again, but just wanted to let you know that we do have a couple things planned for you, and we would welcome you joining and registering for those webinars as well. Allow me to introduce myself. I'm Dr. Leah Dorman, and I'm a veterinarian as well as Director of Communications and Consumer Engagement for Fibro Animal Health. And I'll be serving as your moderator today. Just a few logistic things. All attendees have been placed in listen-only mode, but we do welcome your questions. And one of the ways to ask those questions is at the bottom of your screen. You'll find a toolbar, and that toolbar has a little Q&A button. Be sure and click on that Q&A button as you have questions. Those questions can be typed at any time during the presentation. So you are very welcome to type into that question and answer box the question that you might have for our speaker today. So be sure and do that as the speaker is talking, and we will answer those questions. We will ask and answer those questions at the end of the webinar. Now I do have just a couple poll questions that I'm going to go ahead and launch that poll right now. I'll give you a few seconds to read that. Choose the sentence that best describes you. I am a nutritionist, I am a veterinarian, I am a producer or rancher, or you might be with allied industry or something else. You can choose that other button. I'll give you just about 15 seconds to go ahead and complete that. Looks like we've got several coming in yet, so we'll give you a little bit more time. We've got about 75% of you have voted, so just a few more seconds and we'll share that. And our results, looks like most of you are nutritionists. We've got a few veterinarians as well as a few producers or ranchers out there, and quite a few that are others. Thank you again for joining us. The second question we have for you is complete the sentence. I work mostly with beef cattle, cow-calf, or beef cattle feedlot, dairy cattle, small ruminants, usually sheep or goats, or something else. Again, we'll give you a little bit of time. Got about half of you who voted. Just a few more seconds. Very good, thank you for that. 18% of you are beef cattle, cow calf, about 53, so over half of you are beef cattle feedlot. We've got 26% dairy cattle and 1% small ruminant sheep and goats and 1% other. So thanks again for that information. It's always really helpful to know who we are talking to. Here we go. So we're very pleased to have with us today Dr. Louise Tedeschi. who will be giving us an overview of rumen health and its importance on cattle productivity. I have the pleasure to introduce him today. Dr. Tedeschi is a professor and fellow in the Department of Animal Sciences at Texas A&M University in College Station, Texas. His research expertise is in ruminant nutrition, bioenergetics of growth and development, assessment of feed quality, and modeling and simulation. Dr. Tedeschi has published more than 210 peer-reviewed papers and 30 book chapters and presented at more than 140 international conferences. He has advised 40 PhD and 24 masters and mentored 6 postdoctorates from around the world. Thank you so much for joining us, Dr. Tedeschi. If you want to go ahead and share your screen, you can just take it away from me. And the floor is yours.
Thank you, Leah. Can you hear me?
We can hear you. Excellent.
Okay, thank you. Thank you. So like Leah said, this is the first presentation in this series of Rumen Health. It's really a pleasure to be here talking to you. And I want to talk a little bit about this Rumen Health Compendium. This is a publication that we released now in 2020. It has about 14 chapters and written by 10 scientists from around the world. In this publication, we cover fundamentals of the rumen and anatomy, physiology, rumen development adaptation, rumen microbiome, and contents in the rumen. And then we go on and talk about manipulation of the rumen, pathology, subacute rumen acidosis, liver abscess, bloat, poisoning, and poisoning due to feeding. Then we talk about diagnosis of diseases due to malnutrition. And the last part, we talk about gut, foodborne pathogen, and antimicrobial resistance. This publication is really an advanced rumen nutrition course. It's very illustrated, really well illustrated, and has a lot of references too. More than 660 references for those that want to know deeper about this Rumen Health Compendium. Like Leah said, there will be 3 presentations. My presentation will go on this overview of rumen anatomy and physiology, health and importance for cattle production. And then the second presentation will be subacute acidosis and its impact on liver abscess. And then the third presentation will be on rumen modifiers and feedlot management. So the outline for this presentation today is what are the important aspects of anatomy and physiology for understanding the rumen functions. Then I'll give a brief overview on ruminal diseases and preventions, and those are related to feeding ruminants. Then I'll touch a little bit on rumen modifiers to fine-tune feeding to achieve greater performances, and talk a little bit about diagnosis of ruminal dysfunctions. So let's start with the first topic. This slide has the topography of the thoracic and the abdominal organs of an adult cow. We can see the location of the reticulum right here, and the rumen. All of this structure here is the rumen. And this little bitty here is the abomasum on the right side of the animal. We can barely see it, but it's on the other side. I just want to locate you where this rumen is located in the abdominal cavity of this animal. It's in between this. This is the 4th rib of the animal, A, and B is the 13th rib of this animal. So it's right here, so locate you where it is. Important things here is the reticulum, as I mentioned here. It's this number 1 here. Then you have this reticular groove here. You have the rumen. All of this is the rumen. You have the cranial sac right here. So when I'm talking about this, you know where I am at. The dorsal sac right here. The caudal dorsal blind sac is this number 5 here towards the end. Then you have the ventral sac here, number 7. Then you have this caudoventral blind sac, number 8. And you have all these cranial pillars here, as I'm showing here. Number 9 is the key one here, but you have several others here. And these are very important for rumen motility, as we will see. So the rumen is the largest compartment. It's about 200 liters, and is responsible for fermentation and absorption of nutrients. Fermentation, it's a fermentation vat that provides a hospital environment for obligate anaerobic microbes. And I'm listing here bacteria, fungi, and protozoa. It is a nice place. It provides a nice place to these microbes here because it removes all the byproducts. It controls the temperature and pH, and it also mixes the substrate. So the microbes have a good chance or probability of attaching to the feed particles for fermentation. Papillae is probably the most important structure in the rumen, and we'll talk a little bit more about this in a bit. But it's really to absorb, main function of the papillae is to absorb the short chain fatty acids or volatile fatty acids that are produced during the fermentation, and also electrolytes via passive diffusion. It can be bigger and denser inside of the blind sacs, as I mentioned before, but you have less numbers of those in the ventral sac. This structure here is the rumen, but this is the tail of the animal now, and this is the head of the animal now. So feed comes in here, it mixes here in the rumen until it gets out in this reticulomeson orifice here. This is the reticulomeson orifice. Then the digester reaches the omasum here, number 3, where you have some absorption of liquids. Then it gets out of the omasum through this omasum-abomasum orifice and reach the abomasum, where you have the acidic digestion. It's very similar to the stomach of non-ruminants. The duodenum here, and then it goes onto the small intestine, and then on to the large intestine. And I'm giving you a comparative size of an adult cattle and adult sheep. What is the liters of a compartment in the rumen, the reticulum, the omasum, and the abomasum. And all of these illustrations and information is in that publication I mentioned to you. So let's talk about the movements, the contractions in the rumen. So the mixing contraction is the first one. It starts near the cardia and progress across the dorsal and the caudal regions of the rumen. Then it continues down to the ventral portion of the rumen and to the reticulum back to the cardia region. As the mixing contractions occurs, the digester frequently moves from the reticulum to the dorsal sac, to the ventral sac, to the caudodorsal blind sac, and then back to the dorsal sac. The next one is the eructation contraction. This one initiates in the caudal region of the rumen by contracting the caudodorsal blind sac and relaxing the caudoventral blind sac. The contraction moves forward to the cranial sac, and it's taking this gas cap, as we call it. That's the gas produced in the anaerobic fermentation of the rumen, towards the cardia. At this point, the caudoventral sac, blind sac, relaxes, allowing the fluid level to recede below the cardia, in this slide here, relaxing the lower esophagus sphincter and allowing the gas into the esophagus, right here, the red arrow. The gas is pushed out the esophagus during the inspiration process of air by the animal, causing a portion of the eructated gas to go into the lungs through the trachea. Then there is a problem here. If there is frothy bloat or the animal is lying on lateral recumbency, the cardia will not open because gas contamination by frothy or the digest is sensed by the cardia, and then there is no eructation of the gas. The third contraction is what we call the regurgitation contraction, or the rumination that initiates the rumination by itself. It commences in the middle of the dorsal sac, accompanied by some elevation of the ventral sac that pushes the gas cap towards the caudal region. This is the gas cap and is moving to the caudal region here, and the raft or the mat, which is the fibrous part of the digestion in the rumen, towards the cardium. Upon relaxation of the esophageal sphincter, again, a bolus of the digestive from the ruminal raft enters the esophagus, and it is propelled up the esophagus by antiperistaltic contractions, reaching the mouth, where it is chewed for 1, 2, 3 minutes and eventually re-swallowed by the animal. The frequency and intensity of ruminal contractions are important diagnostic criteria for ruminal and whole animal health, because ruminal and extraruminal factors affect the frequency and intensity of ruminal contractions. The typical frequency is 5 to 10 contractions in 5 minutes in cattle, a little bit less in sheep, but this is about 1 to 2 contractions per minute. The motility frequency does not distinguish between the primary and the secondary or the eructation contraction sequences, which are coupled at a ratio of 1 to 1 or 4 to 1. Means there are 4 primary contractions to 1 eructation contraction. As shown in the table here on the right, some behaviors are stimulatory while others are inhibitory to the reticuloruminal motility. For instance, feed intake, chewing, stretching of the reticulorumen, milking, a low pH in the abomasum, low environmental temperature, all of those stimulate ruminal motility. Others such as bloat, high VFA, they actually have high VFA, high ambient temperature, like in heat stress, low pH in the duodenum, inflammation, ruminal acidosis, fever, satiety, high blood sugar, all of those, they are inhibitory. They tend to stop rumen motility. This table shows the physical on the left and chemical on the right characteristics of the rumen. The range of physical chemical characteristics of the rumen can be quite large, but an ideal operation of the rumen requires a balance among these physical chemical characteristics. Therefore, homeostatic signals are used to keep the rumen within its ideal condition. For instance, the main osmotically active anions are the volatile fatty acids, acetate, propionate, and butyrate, which vary in their molar fractions depending on the diet. Increasing the concentrations of VFA implies that the concentration of other anions, such as chlorine, is low. The anions are electrically balanced by potassium and sodium. If the dietary potassium concentration is high, it will trigger greater absorption of potassium in the gastrointestinal tract to maintain its balance. The total gas pressure in the rumen is almost equivalent to their percentage in the rumen. And the major ruminal gases are in the fermentation are the carbon dioxide, I'm listing 65, but it could vary from 50 to 80, methane, 20 to 40%. They are the main ones. And the small ones will be the nitrogen, oxygen, and hydrogen. And although ammonia is also a gaseous end product of fermentation, it is immediately and almost completely converted to ammonium at physiological ruminal pH values. The anaerobic environment of the rumen has a highly negative redox potential, implying that reducing gases like hydrogen and hydrogen sulfide are generated as minor gases, and that the partial pressure of oxygen is extremely low. The ruminal epithelium can absorb the physiologically relevant cations, magnesium and calcium, for instance, which has attracted a lot of attention concerning the prevention of gas tetany, which is caused by the hypomagnesemia, and milk fever, hypocalcemia, respectively. For the magnesium, the rumen is the main site of absorption, and any deficit in the systemic magnesium homeostasis is a problem happening because of some dysfunctions in the rumen. Ruminal calcium absorption also proceeds to a significant part via calcium channels of the TRP, the transient receptor potential modulators. It is complemented by intestinal calcium absorption, which is upregulated by vitamin D according to calcium demand. The ruminal calcium channels, however, is not responsive to vitamin D, but stimulated by certain plant bioactive lipid compounds, such as this menthol, as an example. Given the intracellular calcium concentration is much lower than those of the magnesium, the driving forces for calcium influx is less sensitive to potassium. Although transporters for amino acids and peptides have been identified in the ruminal epithelium, there appears to be no functional relevant amino acid or peptide absorption in the rumen. It seems that this is a good thing because it spares amino acid and peptides from absorption to preserve those valuable building blocks for microbial protein synthesis. A key feature of a ruminal ecosystem is that the microbiota is not dependent on feed protein supply. In addition to using feed protein, the microbiota can synthesize all amino acids required for protein synthesis using de novo. From non-protein nitrogen sources. The critical feature of microbial metabolism in the rumen is the conversion of carbohydrates to short-chain fatty acids or volatile fatty acids. This conversion is much efficient than glucose is on an only short-lived intermediate with very low concentration in the ruminal fluid. Under intensive fermentation conditions, however, glucose concentration may exceed that value, and relevant quantities of glucose may be taken up by the cell in the rumen epithelium through the sodium glucose co-transport, and also via passive GLUT uniport. Most of the glucose will be used for energy metabolism of the epithelial cells and partly transferred into the blood as lactate across the basolateral membrane. Lactate can be recycled to glucose in the liver. Apart from blood glucose sparing and promotion of gluconeogenesis, glucose absorption regulates glucose availability to microbes and thereby counteracts ruminal acidosis. Because the vast majority of glucose and other fermentable carbohydrates are microbially converted to short-chain fatty acids, the ruminal epithelium has evolved and a great capacity to extract those acids directly from the rumen, which is not only essential for the coverage of animals' energy supply, but also to counteract end products inhibition of fermentation by short-chain fatty acids. The absorption of short-chain fatty acids, or VFAs, is probably one of the most effective pH stabilizing mechanisms for the rumen. In high producing dairy cows, daily short-chain fatty acids production may exceed 100 moles per day, which is equivalent to 7.58 kilos of pure acid. Without effective absorption, the animal could never cope with such an acid load. But how does it happen, and what is responsible for that? So the ruminal epithelium is non-glandular, meaning there is no secretion, multi-layered, and it's composed of different epithelium shown in the figure here on the left. There are four major layers. The first one is the inner portion is the stratum corneum, then the granulosum, then the spinosum, and then the basal. The corneal layer is the innermost layer, and it's in contact with the digestion. It's also covered by the epimural bacteria. It is composed of multi-layered keratinized cells. Keratins are fibrous proteins that are insoluble, unreactive, and durable, which play a role in the ability of the ruminal epithelial surface to resist the abrasiveness of the ingested feed, the digestion in the rumen, and all of the movements in the rumen. The cells have the absorptive ability and also are responsible for the protection of the underlying layers from the contents in the rumen. And that's a very important task. The ruminal wall is not a uniform, smooth layer of cells interposed between ruminal contents and the muscular layer. Instead, the epithelium layers forms projections we call papillae. The papillae are an extension of the ruminal epithelium and are intended to expand the surface area for absorption and metabolism of end products in the rumen. It works as a protective barrier. It does undergo substantial changes before weaning of the animal. And it is a mechanical stimulation, what we call a scratch factor, is very important to help with the development of the papillae. Therefore, the health of the papillae is the utmost importance when dealing with ruminal health. Butyrate, production of butyrate has a stimulatory effect on the development of the papillae, and propionate too. So in the rumen ecosystem, we find microbiome. That's the microbes in the rumen. Rumen bacteria are the most abundant and diverse group of microorganisms in the rumen ecosystem. Ruminal contents can contain 10 to 10 to 10 to 11 bacteria per ml or grams, and which are responsible for the majority of the fermentative activities in the rumen. They possess a multitude of enzymatic activities, including amylases, cellulases, hemicellulases, proteases, lipases, to digest starch, fiber, proteins, and lipids. Most of the bacteria, 70 to 80%, are attached to feed, as the attachment is a prerequisite for the digestion of the insoluble feeds in the rumen. Bacteria are also present in the liquid fraction, from 20 to 30% of ruminal contents, where they utilize soluble substrates and are in transition from one feed particle to the next. A small fraction of bacteria, less than 1%, is also attached to the rumen epithelium, as I mentioned before. Protozoa is the next one. It's present in the rumen fluid at 10 to 3 to 10 to 7 cells per ml, and it may account for up to 50% of the total microbial biomass. Protozoa are thought to be responsible for a quarter to a third of the fiber digestion in the rumen. Although protozoa are often associated with rumen fluid, large numbers may also attach to the surface of the feed particles. Of the ruminal and reticular epithelium, the protozoa are also predators of rumen bacteria. Therefore, the number of bacteria in the rumen fluctuates inversely with the numbers of protozoa. Fungi are the least numerous of the microorganisms in the rumen. So 10 to 3 to 10 to 6 zoospore per ml, and represent less than 20% of rumen microbial biomass. However, rumen fungi are among the most important microorganisms involved in the digestion of low-quality forage, such as cereal straw and bad quality hay. Methanogens are members of a unique group of microorganisms known as the archaea, and are responsible for methane production in the rumen. Methanogens are not prominent members of the microbial community, 10 to 6 cells per ml, but their ability to reduce formate and carbon dioxide to methane is an important component of the fermentation. Bacteriophages are bacterioviruses. that reside in the rumen in concentration of 10 to 10 particles per ml of rumen fluid. These viral particles need a host bacteria for replication, which may culminate in rupture of the bacterial cell and end up killing the bacteria. Bacteriophages are extremely host specific, often being able to infect only a few strains of a given bacterial species. To date, more than 125 different morphological types of bacteriophage have been identified. So the rumen is, as I mentioned, inhabited by a microflora, but modern nutrition models, such as the rumen nutrition system, has only 2 pools of bacteria. The FC bacteria ferment fiber, and the NFC bacteria utilize non-fiber carbohydrates. These 2 types of bacteria have fundamental differences in terms of nitrogen metabolism. The FC bacteria uses ammonia as the nitrogen source and do not take up amino nitrogen and never produce ammonia. The FC bacteria, on the other hand, the NFC bacteria, they can use ammonia, but they prefer amino nitrogen like peptide and amino acids to grow. In the case that the rate of the NFC carbohydrate degradation is insufficient to drive the protein synthesis, incoming amino acids will be degraded and converted to ammonia. The growth rate of both types of bacteria is dictated by the rates of degradation of the fiber and then the non-fiber of carbohydrate. The Rumen submodel uses growth rates to predict the amount of energy that is available for growth and the amount that is expended on maintenance. Ruminal bacteria can also dissipate energy in futile reactions that are called energy spilling. The NFC bacteria spill energy if they're deprived of amino nitrogen or the ammonia nitrogen, and the KD is faster than the growth rate, or the KD of degradation of this peptide and amino acid is greater than the source of NFC of the diet. So this slide shows the requirements of bacterial growth in terms of nitrogen sources. So for the ones that ferment non-fiber carb, there is a need of 67% for amino nitrogen and another 33% coming from ammonia nitrogen. Whereas those that degrade fiber, they need 100% of their nitrogen is from ammonia nitrogen. And then this explains what we mentioned in the previous slide of the uptake of amino nitrogen. If there is any energy spilling, if the amino nitrogen balance is greater than 0, if it is not greater than 0, then that is an energy spilling. If it is greater than 0, that is a deamination, and so forth. And all of this is produced here. That's how it is used in the rumen. In general, a healthy rumen, these carbohydrates are fermented to volatile fatty acids by the rumen microbiota at different rates and to varying degrees, as shown in the figure here on the left. So, single sugars are the most rapidly degraded of carbohydrate within the rumen and are generally completely fermented by the bacteria within minutes or a few hours. Non-structural and structural carbohydrates are more chemically complex and are often encased by other compounds such as protein and lignin. Therefore, their degradation is not as fast as simple sugars. If cattle are fed high-grain diets, for instance, amylolytic bacteria like Ruminobacter amylophilus that digest the starch, and those that utilize the end products of starch digestion, such as Megasphaerus danii, they use lactic acid predominate in the microbiome. This change in the microbiome leads to a decrease in acetate and an increase in propionate proportions. The production of more fermentable and end products leads to a decline in pH. As I'm showing here, pH is going down. if the production of a more fermentable end product leads to a decline in pH, and if it reaches 5 or less, lactate concentration in the rumen usually increases. At a pH below 5.8, fibrolytic bacteria are inhibited. Therefore, fermentation of fiber decreases drastically. So given all of this basic information on anatomy and physiology in the rumen, what are the key diseases and preventions? So let's talk with rumenitis first. To me, rumenitis is the beginning of all problems. It's an inflammatory change that develops in rumen wall, epithelium, and underlying tissues. And I'm showing here, this is the rumen wall, and those are ulcers in the rumen wall, the epithelium. It's commonly observed in cattle-fed high-grain diets because some of those are inadequate in forage or fiber or physically effective fiber. So you have a congestion, then a hemorrhage, edema, bacterial and fungal invasion, ulceration, and scar tissue formation. This is the adequate scratch factor. When you have adequate scratch factor or fibrosity in the diet, the length or the size of the papillae is very homogeneous and consistent and really nice. When you're lacking this scratch factor, it's an inadequate amount of fiber in the diet, or PNDF as we call it, the length of the papillae is different. It's not homogeneous. It varies a lot, and even their shape is not consistent. Of course, this impairs absorption of short-chain fatty acids in the rumen. This graph here shows that inadequate scratch factor will result in rumenitis. The absorption and metabolism are impaired. Accumulation of organic acids will lead to acidosis. And the protective barrier function is compromised. There is a penetration by feed particles and hair, and even microbes like bacteria and fungi will cause rumenitis and liver abscess. This graph here, this on the right, shows what happens in the rumen. So this is anaerobic fermentation producing short-chain fatty acids. They get absorbed in the epithelium, go through the portal blood, reach the liver, they're converted, they're processed here in the liver, and then used by the animal. So these are pictures showing different degrees or levels of rumenitis in the epithelium of the rumen. So A is more like a serious fibrous type of rumenitis. B is showing erosion or ulcers, ulceration. Those are these red marks here in the rumen wall. This one is a scar. There was an ulcer here right at this point, and then once it closed, it healed, you have all this tissue here that has no papillae attached to it. Therefore, all of this region here has no absorption potential of short-chain fatty acids. This one is hemorrhage. Is showing from the outside of the rumen. So it's showing these red marks here. It's problems from inside of the rumen. This is how to prevent rumenitis, adequate forage, roughage to provide the scratch factor that I mentioned before, prevention of suboptimal ruminal pH via feeding and management is totally adapted to what we call diets, a diet with high concentrate. Inclusion of buffers, bicarbonate, direct fed microbials, ionophores, antibiotics, and others. It's a good idea. And I call for this a smart use of feed additives. And I'm giving you some examples of rotational, combinatorial, and strategic. So using these ways here of feeding feed additives might be a good way to decrease rumenitis in the rumen. Liver abscess is the next one. I'm just going to show you a little bit of the liver abscess as it will cover next in the next presentation. It's also called known as a silent infection. There is no clinical signs, Blood liver functions tests are normal. There is no antibody titers. And ultrasound imaging is very hard. It's sometimes not accurate unless you have very good quality imaging, ultrasound imaging system. Reduced animal performance, as you can imagine, because all of this damage in the liver. Reduced intake, weight gain, feed efficiency, carcass weight, yield, and no liver value. This picture, this graph here shows different pictures of different levels or degrees of ulcers in the liver, even inside of the liver. This one is looking from outside of the liver. You see these marks here are abscess from, going outside on the outside of the liver. Really, really bad situation. Those are the scoring system that you use. This one is a normal liver. There is no abscess. We don't see any abscess here. Then we have this 1 or 2 small abscess or inactive scars. We call it A-. Then you can have 1 to 2 large abscess or multiple small abscesses. It's an A score. And of course, imagine the A+ is multiple large abscesses. This is really bad. How bad it is? It decreases as you increase those degrees of abscess in the liver. It decreases the hot carcass weight by a lot. And here I'm showing you it's a significant reduction in hot carcass weight. Subcutaneous fat depth decreases. The longissimus muscle area also decreases. The yield grade decreases, and even marbling score can decrease depending on the intensity of those A+ that you get. This is what we believe happens. This is the acidosis, rumenitis, and liver abscess complex, as we call it. High grain feeding in the rumen, causing acidosis, impair the rumen wall or the epithelium, causing rumenitis. Bacteria will go through the rumen wall, escape the rumen. They will reach the liver, and they will reside in the liver and cause liver abscesses. It's essentially, this is a big picture of what we think it happens. Who causes it? Fusobacterium necrophorum is the most prevalent one. It ferments lactate to acetate propionate and butyrate. It's a proteolytic bacteria. Major lysine fermenter, increases with grain fed and also cause other problems. It's a normal bacteria in the rumen. It's always there, but it's not the only one. Many bacteria can cause liver abscess, and I'm showing here several of those that can cause problems. Prevention for rumen abscess, liver abscess, sound feed bunk management, antimicrobial feed additives, antibiotic and antibiotic alternatives like bacteriophages I mentioned before, vaccines. And reasons for incomplete control of rumen liver abscess. Source of the Fusobacterium is we're not really sure where it's coming from, rumen hindgut soil. And when the cattle is developing the liver abscess, if it is during the growing phase, the step up, the finishing, or even closer to the slaughter. We don't know. We don't have complete understanding of these factors here, though starting to measure some of those. The next one is rumen acidosis. I'm not going to cover much of this. I'm just going to brief talk about this rumen acidosis because it is important to understand what is going on. So in the rumen, there is this normal fermentation of the substrate in the rumen. And I'm just showing here this dynamic fermentation of different substrates in the rumen, and it produces acid in the rumen. Those acids change, and it depends, the production or the peak of those acids depend on how often you're feeding these animals, how much those animals are consuming, and so forth. The problem with this is how long it stays in the rumen. It's not really much about how much is produced per fermentation, but how long does it stay in the rumen? It stays in the rumen depending on the passage rate, or how fast it gets out of the rumen, or how fast it gets absorbed from the rumen. And I'm just showing you here the fractional absorption rates of these acids, and it increases when you have a lower ruminal pH. This is absorption.
My apologies for interrupting. Some people are having some issues with their audio. And if you are, there are some instructions in the chat box in how to remedy that. You can switch to phone audio by clicking on the arrow next to the microphone and switch to phone audio. And the details are there. The details are in the chat box. My apologies, Dr. Tedeschi, please continue.
Okay, Leah, okay, thank you. So this is absorption of lactate. When you compare to other VFA products in the rumen, the absorption of the fractional absorption rate of the lactate is lower than the ones you have in the VFAs in the rumen. That's why you have accumulation of the lactate in the rumen. So something we can think about is a physically effective fiber like the PE NDF. You increase the rumination activity, you have saliva production, it stimulates saliva production, forms the rumen mat, stimulates ruminal motility, passage of material through the gastrointestinal tract, protects the rumen wall by maintaining the buffering capacity, reduces the depression of milk fatty for lactating animals, and reduces the pressure of the cell wall. So the next one is bloat. Two types of bloat. The first one is free gas. So you have this impaired ruminal motility due to nerve disorders. Usually it's the vagus nerve. And the other one is frothy bloat. This is a frothy bloat picture here on the left. Right on the left, the right picture. Caused by pasture, most likely due to soluble proteins. In feedlot, it's because of this excessive production of mucopolysaccharide by the bacteria that creates this viscous fluid preventing separation of the gases. So this one shows you a picture of traumatic reticuloperitonitis that can cause bloat, ruminal acidosis, bloat here in this picture, or simple indigestion or vagal indigestion. In case of acute frothy bloat, passage of a stomach tube seldom produces dramatic relief of the rumen distension, but does aid in diagnosis and allows treatment with a surfactant agents such as product based on poloxosaline drenches, or vegetable oils to reduce down the froth. Emergency ruminectomy, like this one I'm showing here, is the treatment of choice for progressive and severe frothy bloat cases. But is it gonna happen again? Maybe we should improve better feeding of these animals. This one showing different pictures of a severe distension of the rumen. And this one is a free gas bloat. For the pasture, avoid those pastures that cause bloat, like these ones I'm listing here, the moderate risk or the low risk ones here. It can become really bad. And treatment strategies mentioned here for feedlot bloat, most of those are linked to acidosis, and some of those are linked to pasture bloat, can be mitigated by selecting which pastures you're feeding to your animals. Quickly about rumen modifiers. So there are many rumen modifiers that enhance the beneficial processes in the rumen, minimize or inhibit inefficient processes, or minimize or inhibit harmful processes in the rumen. It has, there are many feed additives and rumen modifiers, like this one are hormone-like, ionophores, or antibiotic. A list of ionophores, and all of this is in that publication I mentioned to you in the beginning. All of this started back in 1920, and it's been discovered new ones and different use of this. I want to touch a little bit on this virginiamycin antibiotics produced by this Streptomyces virginii. it reduces the incidence of liver abscess because it inhibits the growth of this fusobacterium. It increases the animal growth because of the effective reduction in lactate and possible increase in propionate concentration in the rumen, besides sparing ruminal protein from deamination. In the beginning, some claims about a reduction of acidosis was done. And in this study here, they fed Holstein steers. This one is a basal diet of alpha hay and concentrate, and they induced acidosis withholding feed for 24 hours and then adding a slurry of powdered corn and corn starch, a huge amount on a daily basis. As you can see here, the ruminal pH was greater, the top one showing ruminal pH, at times 6, 3, 6, 9, and 12 for VM, and did not differ from monensin and Thailand after 30, 51 hours. But virginiamycin was also able to lower the lactate concentration for the first 36 hours compared to control or the MT monensin Thailand treatment. So in this experiment here, this is another analysis we did with more than 7,000 animals. We performed the meta-analysis on what is the effect of virginiamycin. And here showing virginiamycin different concentrations here on the x-axis, liver abscess incidence, and average daily gain here on top. We found that between 12 and 24 milligrams per kilo of dry matter was the most effective to reduce liver abscess. And we actually found that when it was fed above 18 milligrams, there was even higher probability of observing statistical difference of liver abscess incidence compared to control animals. We believe the maximum reduction might be achieved if we go beyond the 24 milligrams of VM per kilo of dry matter. As we can see here in the plot, that's exactly what this plot is showing after 24. That is a huge decline here in the liver abscess. So quickly, to finish up diagnosis of dysfunctions and disorders, these figures are in the publication I mentioned to you. This one here is looking from the behind is a normal animal. If you see a bump here on the left side, it's more likely free gas bloat. This is what happened in the room. It's happening in the rumen. Both sides frothy bloat. On top, on the left here on top, and on the right here lower part is a most likely vagus indigestion. This is fecal scoring, another a tool to look at what is happening. Most likely acidosis on A in the rumen, most likely some lower pH in the hindgut, and this one is probably a shortage of a protein. And the third presentation, we'll talk more about this. You can go over some of these scoring, fecal scoring systems, 1 to 5, all the way to not enough protein to too much protein. So in summary, I think the success in the production of the ruminants depends on how we control the dynamics of fermentation in the rumen and how we feed the microbes. And preventing that ruminite is key. So dysfunction disorders caused by the lack of complete understanding of management of the rumen nutrition is most likely what will happen. Rumen modifiers, mode of action, include changes in fermentation metabolic pathways, modifying the microbial ecosystem in the rumen. Their efficacy depends on the type of feed, physiological status of the animal and animal species. And most important here, as I mentioned before, is what type of strategic feeding can we use combining different rumen modifiers to improve fermentation in the rumen. So this is just for you to think about. Our mom tells us, wash your hands before you eat to remove the germs. That's very normal for us, non-ruminants. In a ruminant world, would probably say the other way around. Watch out what you eat. You have a belly full of germs. So you need to know what you're eating because you want to keep those germs in the belly controlled. So at this time, be glad to answer questions. And Leah, I'll transfer back to you, I guess. Thank you.
Thank you, Dr. Tedeschi. I think it's always important to listen to your mother. I think I definitely heard that, or maybe I just need to share that with my kids. Hey, we've got a fair number of feedlot folks here, and one of the questions, and just a quick reminder, if you do have a question, go ahead and type that question in the Q&A box, which you can find in your toolbox as you hover over it. So probably at the bottom of your screen if you have a PC, but you can type those questions right in that Q&A box. But a question about feedlots. In feedlots, what is the period at which the risk for acidosis is the greatest?
Yes, we're actually looking into that. That's a very good question. Leah, can you hear me?
I can hear you just fine. Yeah. Yes, thank you.
Okay. So yes, it's a very good question. I think the sense of the what everybody believes is during the transition phase from the finishing from the growing to the finishing phase, we think might be the critical part of causing acidosis, mainly in the beginning of the finishing phase might be that. But it also depends on what is fed during the growing phase and what is the proper amount of fiber and things like that. But I think it's a very good question. I would say it's really during the transition phase. It's the main one, Leo.
Great. Another question we have, Dr. Tedeschi, under practical conditions, what is the best way to fully control acidosis, both clinical and subclinical?
I think it's a practical way. I think it really depends on what is your target of production, what you're trying to get out of your animals, and for how long you're trying to feed this. I would say I think it's maintaining the minimum fibrosity of your diet in terms of diet formulation of these animals. Guidelines, and I believe that Dr. Jennings will probably talk more about this. So the guidelines that I'll say is something like this. Don't ever go below 7% of PE NDF. And if you want to improve performance, I would say stay above 10 to 15%. That's what I'll do. Use your feed additives. Follow the recommendations on the feed additives. And do a good transition. Slow transition, to me, I think it's the best one is in preparing those animals for that, for the hot diet, for the finishing diet.
Thank you very much. Someone asked, the efficacy of sodium bicarbonate is controversial at rumen pHs lower than 5.7. What is your opinion on this? Yes,
I think it's a lot of research there actually showing this sodium bicarbonate. I think that the problem with that is this: you have to make sure that those animals have a consistent consumption of a sodium bicarbonate to actually control the ruminal pH. But the problem with the ruminal pH is not really controlling the buffering capacity of providing sodium bicarbonate, for instance, or even calcium carbonate or something like that. The main problem really is making sure that the animal can actually absorb that short-chain fatty acids or the VFA to actually decrease how often, how long it stays in the rumen. Because once it stays in the rumen and starts causing problem like the rumenitis, then it's gone. The sodium bicarbonate is not going to be effective at all. And the problem with the sodium bicarbonate is also this: how fast does it escape out of the rumen, and what is the effectiveness of it in the rumen, essentially.
Great, thank you. But there's another question about virginiamycin versus sodium monensin, which controls better ruminal acidosis. And I'll have Dr. Milton jump on here as well. So Dr. Milton, if you can unmute and show your video, we'd love to hear your answer as well. But Dr. Tedeschi?
You want me to start or Milton? I can start.
Okay, yeah, go ahead.
And I think we showed that in our publication. We did show in our publication back in 2018. I think the virginia-mycin had the amount that we had to feed of virginia-mycin was a lot less than we had to feed of a monensin, for instance, to reduce ruminal acidosis. I think it might depend on the type of the diets you're feeding or something like that, and how fast you want to decrease the ruminal acidosis, something like that.
Yeah, and if I can follow up on that, I think that one of your comments was on the strategic use of the different technologies that we have available for controlling acidosis. And I think that when we understand how the two products work, they truly work complementary. So one will achieve a better control of rumen fermentation through the control of feed intake, that's monensin, whereas with virginia-mycin, you may have an even stronger effect on those lactic acid producing bacteria, as the trial that you showed during your presentation. So I think that both products are very effective, and I would just further subscribe to your comment that through the strategic use and whenever it is possible, I think that it definitely makes sense to utilize both products to achieve the best results.
Thanks to you both. Getting a question also about rumen modifiers. Do they work better or worse during different stages of the feeding program? Any thoughts on that?
That is a good question, and that's what I tried to allude to when I was talking about those three different ways of complementary feeding of rumen modifiers. The combination of them, the strategic, which is which one comes first, and which one comes second, and vice versa, and rotational. Even if you feed one first and then you come back to, you go to number two, then you come back to number one, and so forth. I don't think we have a clear answer on which one is the best one, but I'll tell you for now that there are some publications out there showing some benefits of a rotational of different products. I don't think we know a lot at this time to give you a clear answer of which ones and when, but definitely I think they'll be beneficial, because I see a complement of feeding different rumen modifiers.
Thank you. An easy question, your book, Rumen Health Compendium. How do you get a copy of that?
That is a very good question. You need to talk to your fiber representative. Make sure that you call them up as soon as we're done here, and to make sure how you can get a copy.
Thank you. I'm just scrolling through some of the questions. We are actually out of time, and as much as I want to answer all of them, if you want to stay for just a few minutes, we do have just a couple short announcements, and then we can get back into the questions if you'd like. But I do want to at least say, first of all, thank you so much for joining us in this first of a series in the Rumen Health webinar. You can come back to us by all means at academy.pahc.com and sign up for the next couple webinars that we have in this series on May 20th and June 30th, or June 3rd, I'm sorry, May 20th and June 3rd as a reminder. If you have questions about today's webinar, feel free to contact Dr. Milton. His email address is listed there, and Danilo as well. For general information, you can always visit our website. But again, if you're willing to stay for just a bit, happy to go ahead and ask some additional questions here. And Milton, jump in, because oh, there we go, I can flip my screen. You mentioned strategic feeding. Can you tell us what that means? What do you mean by that?
Yes, a strategic feeding would be exactly when do you start feeding different diets, and when you add different modifiers, and what is the amount that you enter there? Something about strategic feeding could be, when do you change the composition of your diet? When do you add more protein? When you decrease protein? And also is, how much is your animal really needs? All right, is it the animal in the growing phase? Is the animal in the finishing phase? Which product is gonna benefit the best given the type of the diet that I'm feeding? If it is a, let's say, a hotter diet, if it is a low protein diet, a high protein diet, it depends on what type of diet I have and what type of products I have. It can actually include some of a, let's say, like a condensed tannins or anti-nutritional factors or something like that. So what type of rumen modifiers should I include to prevent some of the negative effects of these products?
Thank you for that. How effective would you consider prebiotics in stabilizing a ruminal pH? And is there a difference among yeast or molds and live against fermentation extracts?
We've done some work on probiotics. We haven't really done a lot on prebiotics. I think the problem is, I think there are too many factors that can affect what is happening in the rumen. And it's we need a very, very controlled experiments, very homogeneous animals, and making sure that everything is consistent and controlled, so we can actually see the real benefits of some of these products. I believe that there might be some important aspects of these products, and but I think we still need to work on how to minimize some of the variation of these products. Those products may actually be they are very good candidates for combining with some of the rumen modifiers that I mentioned before.
Another question, grass feeding and Virginia mycin, do you have any tips?
From the data that I've seen, I think it's very good. I think it works. I don't know what type of tips you want, but I think it works. I think it has its own niche there for grazing animals. It may be a little bit different than for feedlot situations, but I believe the people from Fibro can actually provide you some information about that. We do see some increase in animal performance and of grazing animals. And there might be some improvement also in the rumen fermentation of these animals.
And if I may jump in here, Leah, just quickly to encourage everybody to go and reach out, especially to our colleagues in Brazil. We've conducted several trials and very intriguing information with different supplementation tactics with in very different conditions, like dry season versus rainy season, with protein supplements, protein and energetic supplements. So to summarize all those results, I would say that definitely we've seen a very consistent effect in growth promotion, in average daily gain, and we explain that through an improved fermentation pattern, as Luis was mentioning. So I would just encourage everybody to reach out and to either Danilo or myself, and we can certainly share some of the specifics on the research available.
Very good. Another question, is there any way to recover rumen papillae from acidosis? And how long would that take?
It's like, remember the pictures I show you? I think it depends on the damage. I think if you already caused the damage, like those scars that made in the rumen, then you're lost. It's gone forever. if it is just for a short period of time because you had a shortage of those scratch factors, or it was so then they may change the length, they may change their shape, and then as soon as you feed back a good source of fiber, they will come back up. But if the rule of thumb is this: if you lose them because of acidosis or something like that, and they're gone, or because you have a scar or something like that in the rumen, they're most likely going to be gone. They're not going to come back up. But if it is just a reduction in size and shape because of a temporary impact of the diet, then yes, you can actually bring them back up. As a matter of fact, this is normal in the wildlife and other ruminants. They actually do change the size of their papillae throughout the year, depending on what is available for them and what type of short chain fatty acids it's produced, given the feed that they consume throughout the year. So it is normal to have those shape and size and length changing. But if you damage the rumen wall, most likely you will not get a papillae regrowth.
I think we have time for maybe one more question. I know we've gone a bit over time, thanks to those of you who wanted to stay and listen to some of those answers. So thanks for doing that. Simple sugars spikes are used up quickly in the rumen. If molasses is being used as a carrier for urea, does this affect the pathway of the nitrogen as opposed to other delivery methods for urea in pasture-fed animals?
If it depends, if the molasses is being used as a carrier for the urea or the nitrogen form, as long as it gets solubilized in the rumen, that nitrogen will be used. That non-protein nitrogen urea, whatever you combine it with the molasses, will be used in the rumen. If there is a permanent binding of the molasses or transformation of the sugar, if you make it an alcohol, if you do some sort of a chemical reaction, and then that binds to the nitrogen of the urea, and then it does not become soluble in the rumen, then it's lost, then it's not going to be used. It may even be that bacteria may not even be able to degrade that product, whatever the product become, depending on the treatment you do. But as a rule of thumb, if you're just using it as a carrier, it's a very simple mix. Then once it gets solubilized in the rumen, both will be used normally by the microbes in the rumen, and quickly.
I think maybe one more. For feedlot bloat, is there a proportion of animals that never get used to high caloric diets independently of the type of diet? In other words, what is the importance of the individual susceptibility?
That's a good question. I think Dr. Penner would be very, very glad to answer that question. Mainly because I have no clue if that is a number. I don't remember reading anything about it. I don't know. I cannot even give you a guess. Hopefully everybody will be okay, but we know maybe 3%, whatever, or even 10%, they're not gonna adjust.
I will tell you that Juan Manuel asked that question, and so we hope that you will come back and join us for Dr. Penner's webinar, which will be held on May 20th. So we hope you'll come back to Fibro Academy, academy.phc.com, and that's a perfect segue into ending our webinar today. Again, we thank all of you from Fibro Animal Health. We thank every one of you for joining us today. Please stay safe and stay healthy. And again, have a wonderful rest of your day. Bye-bye.
Thank you.